Hello, my name is Joshua Mugabe. I welcome you to Heredity. Genes control the characteristics of living organisms. What we call the genes, they control the characteristics of living organisms. Let us try to look on our diagram here. We have chromosome A and this is chromosome B. They are called homologous pair. Why? Because they move together. One chromosome comes from one parent and another chromosome comes from another parent. So when they come together in the zygote, they will move together. And on this chromosome, there is what we call the genes. For example, here is capital G, which is a gene for color of the pond. This is a gene on one chromosome, and this one is another gene on another chromosome. Both of them, the chromosomes, we call them homologous chromosomes, moving together and controlling the same character. So the genes are the ones that are going to control the characteristics of a living organism. Now, the characteristic that we are looking at of this living organism is the color of the pond. The color of the pod is what we are talking of as the characteristic of living organism. The genes, these ones are carried on chromosomes. So our genes, for example, this is a gene and this one is also a gene. These genes are carried on chromosome. So this gene here is carried on chromosome A. And this other gene is carried on chromosome B. And we have seen that the genes are the ones that control the characteristics of living organisms. So they are found on chromosomes. The chromosomes are in pairs, one from each parent. Here we have two chromosomes, which we call a pair of chromosomes. This pair of chromosomes, one comes from one parent and another chromosome comes from the other parent. Let's say chromosome A came from the mother, then that means that chromosome B came from the father. So they are a pair, each coming from one parent. Genes are always in pairs. If you look at our genes here, this is a gene located on that chromosome, and this one is also another gene located at that point on that chromosome. So the genes which are controlling a particular character, for this case, we are having a gene controlling the color of the pond. They are always in pairs. They are two, each on its own chromosome. And they are the ones that are going to determine the characteristic of that individual or that living organism. The genes controlling the same characteristics, they occupy an identical position on the corresponding chromosome. So these genes, which are controlling this character, which is the pond color, they are located at an identical position on the corresponding chromosomes. This is a chromosome that is coming from the mother, and this is a chromosome that is coming from the father. But the gene that controls the pond color is located at this place. Even the other one will also be located at the corresponding place, what we are referring to as they are occupying an identical place. Now you'll find that the same chromosome has also genes which control other characteristics. And also those genes will be on the corresponding chromosomes, and they will be occupying the identical position. If we say now this one is controlling eye color, even this one will be controlling eye color. So they will exist in a pair and at an identical position. The gene pairs control one characteristic. Every gene pair controls one characteristic. For example, we are looking at this same position. There is the same gene, and that gene is controlling the color of the pond. So this pair of genes controls only one characteristic, and this characteristic is the color of the pond. It's what they are controlling. But they do not always control it in the same way. It is not that these two genes are going to control the characteristic or the character in the same way. For example, when you look at this one from gene A, it is a gene for green pod. And when you look at chromosome B, this is a gene for yellow pond. So this one is saying that the ponds should be yellow. And this one is saying the ponds should be green. So even if this pair is controlling one character, which is the color of the pond, they do not always control it in the same way. It doesn't mean that when this one says green, this one will also say green. They may be controlling the characteristic in different ways. For the gene pair which help determine the coat color in the mice, one might produce black fur, and its partner might try to produce brown fur. So when we are looking at the gene pair, for example, here we have a gene pair, which is capital B, meaning that this gene is on one chromosome and this other gene is on one chromosome. Then again, we have another gene pair here. This gene, small b, is on one chromosome, and this other small b is also on one chromosome. And all of them, they are on different chromosomes. So, we are saying that the gene pair which help to determine the coat color in the mice, one might try to produce black fur. For example, if you look at our mice here, or mouse, it has a pair of gene, which is capital B, and this pair is producing black fur. And then we have this gene pair here, small b. This gene pair is trying to produce the brown fur. So these gene pairs, they are producing different types of fur. One is producing brown, another gene pair is producing black fur. The gene for black fur is dominant to the gene for brown fur. So here we are having gene B, capital B, is a gene for black fur. And we are saying it is dominant to a gene for brown fur. And the gene we have for brown fur here is this small b. So which means that if this one comes into combination with this one here, and they are together, that means we shall have capital B and we shall have smaller b. If those two genes come together, capital B is dominant to small b. And this capital B is for black fur. And small b is for brown fur. That means that now this combination we are having here, because capital B is dominant to small b, that means that we shall have black fur. The genes are represented by letters. As we are seeing here, this is a gene represented by letter, and this one is a gene. The two, for example, that form the black fur, here is capital B. We shall have the black fur. When gene small b and gene small b are appearing together, then we shall have brown fur. So the genes are represented by letters, capital B and small b. The gene for the black fur is given by the letter capital B. This one, gene capital B, is a letter or a letter representing a gene for black fur. And the gene for brown fur is given the letter small b. Now we have this small b is representing a gene for brown fur. The genes must have the same letter, but the dominant gene is always in capitals. So when you look at our genes here, they have the same letter. And even these genes here, they have the same letters. But for the gene to be dominant, it has to be represented by a capital letter. When we represent the gene with a capital letter, that indicates that that gene is dominant. Then therefore that means it is the other way around. If the gene is represented by a smaller letter, that gene is a recessive gene, and it is going to be written in small letters. The genes must have the same letter, but the dominant gene is always in capital. So like for example, here, our gene for pod color has 2 letters, and they are the same letter, but both of them are dominant genes. Therefore, both of them are represented by capital letters. The genes for the corresponding pair are called alleles. Now we introducing now a new term, which is allele. An allele, these are genes of a corresponding pair. So here we have gene D and gene D. Both of them are genes controlling green color. So we call them alleles as genes of corresponding pair. Genes of corresponding pair. We call them allele. This one controls the color of the pond, and this one also controls the color of the pond. But the way they are controlling may not be necessarily the same. That's why we call them alternative form of genes. We can say these alleles are the same alleles. They are controlling the same characteristic in the same way. But if this one was a small g, there will be alternative forms of gene which controls the pod color. This one will be controlling it different, and this one will be controlling it different, but they are controlling the same characteristic. This means alternative forms of the same gene. Alternative form of the same gene, meaning that it can be allele and allele, but they are the same form. But it can also be capital G and small g, meaning that they are alternative form of the same gene. Same gene means that it is controlling the same characteristic. Alternative form means that it may not be controlling the characteristic in the same way. Capital B and small b, these are alleles for the gene for coat color. So we have this gene capital B and this one capital B. Then we have this gene small b and another small b. They are alleles. They are alleles for the gene for the coat color. They are alternative form of genes for coat color. Capital B is the dominant allele. Wherever we are seeing the capital B, that capital B is a dominant allele or is a dominant alternative form of a gene. And the small b is the recessive allele. This small b is a recessive form of gene. So these two, small b, they are alleles, but they are recessive alleles. A black male mouse, which is represented by two alleles, capital B and capital B, is mated. Mating is the same as crossed with the female brown mouse, smaller b and smaller b alleles. If we look at this mouse here, this is our black mouse, and it has got two alleles, capital B and capital B. Then down here, we have our brown mouse. And this brown mouse has got two alleles, smaller b and smaller b, which are for the brown color. So, in gamete production by meiosis, the alleles are separated. So let us say that we are going to produce gametes. And if we are to produce gametes, here up, we are going to produce two types of gametes. One gamete is going to have allele capital B, and another gamete will also have allele capital B. Then down, we are going to produce a gamete with recessive B, smaller b, and other gamete with allele smaller b. So during the formation of gametes, the mother sperm cell undergo meiosis, and the mother ovum cell also undergoes meiosis, and they end up separating the alleles. So each allele will go in its own gamete. Each allele will go in its own gamete. And that is what we call meiosis. Now, when you look at the sperms that we have produced, this one has got allele capital B, and even this one has got allele capital B, meaning that they are the same allele. And if you look at the ovum, you will see this one will have small b, and this one also has small b allele. So they are the same. So now here we shall take only two of them and combine them. If we take this sperm combining with this ovum, then we shall produce this zygote after fertilization. And you will see that now we shall have a combination of sperm capital B, which is coming with capital B allele, and the ovum small b, which is coming with allele small b. And after fertilization, the two are going to unite, and we shall have a zygote with now allele capital B and small b. These are the individuals that we are going to get. All offsprings will be black. Why? Because they have got allele capital B, which is dominant, and allele small b, which is recessive. Therefore, the presence of a dominant gene is going to suppress the activity of the recessive gene. The recessive gene is indicating that the coat color should be brown, and the capital B is indicating that the coat color should be black. So which color is going to be possessed by the offspring? Here, the color which is going to express itself is the one which is dominant. And when you look here at our combination or pair of allele, capital B is dominant. Therefore, it is the one which is going to express itself, and it will suppress the smaller b. So all the offsprings are going to be having black colored coat. Sperms will carry one copy of the B allele. As we are seeing here, the sperm has capital B allele, and even the sperm also has got capital B allele. The ova will carry one copy of small b allele. So when you look here, the ova we have here, it has one allele for small b, and even this ova we have here has one allele for small b. When the sperm fertilizes the ovum, the allele capital B, which is coming with the sperm, and allele small b that comes from the ovum, they come together in the zygote. So these two, one coming from the sperm, allele capital B, and one coming from the ovum, they are going to unite together and they are going to produce a zygote, which has got now a combination of allele capital B and allele small b. The offspring from this cross is called the F1 or first video generation. So these offsprings that we are getting from this combination, we call them the F1 generation or the first filial generation. They are all black because the allele for black coat color is dominant to the allele for the brown coat color. So we have seen that here they have this allele which is capital B, which is dominant to this allele, which is smaller b. And because capital B is dominant, therefore all the offsprings are going to have the color instructed by the dominant allele. All of them now will be black coated color. The capital B, smaller b mice are called heterozygotes because the B capital and B small alleles have different effects, producing either black or brown coat color. So our zygote, which we have got, it has got the capital B allele, which instructs the coat color to be black. And then we have also small allele B, which instructs the coat color to be brown. The mice are heterozygous for coat color because this one, capital B, is instructing the color to be black and smaller b is instructing the color to be brown. So they are different alleles. So we call them heterozygous for coat color. They have got two alleles which are giving different effects to the characteristic of the mouse. Capital B mice are called homozygotes because the two alleles, they produce the same effect. Now here, these ones we are going to call them homozygous. Why? Or homozygotes? Because the capital B allele and capital B allele, they have got the same effect on the coat color. The small b mice are also homozygous for coat color. Both alleles produce brown coat color. So when now we come to small b alleles, both of them, they are also producing the same effect. And therefore, we call them homozygous also for coat color, because both of them, they instruct in the production of the brown coat color. The figure shows what happens when two heterozygotes are mated and they produce young. So let us study this structure, this diagram figure very well. We have now heterozygotes. Our heterozygotes, they have the same allele. Capital B, smaller b. And here we are having capital B, smaller b. So we said when this one happens, we undergo meiosis. So this one will produce the sperms, one with capital B allele, another sperm will have smaller b allele. When it comes to the ovum mother cell, this one is going to produce an ova with capital B allele, and it will also have another ova with small b allele. So those are the gametes that are going to be formed, which means that there is going to be different combination. We have produced two sperms, this one with allele capital B and this one with allele small b. And we have produced two ovum or ova, this one with capital B and this one with small b, meaning that this sperm can have a possibility of going with this ovum, or it can also have the possibility of going with this ovum. But let us start with the possibility of capital B sperm going with capital B ovum. So they will give us now this offspring up here. When they are combined, we shall get capital B, and we all of us know that capital B allele, all of them are dominant, therefore all of them, they are going to express themselves and the offspring will have a black color. That is when this capital B has gone with this capital B. Now let us look at the capital B going with a smaller b. So we shall end up getting this offspring here. This is a capital B allele which is coming from the sperm combining with the smaller b which is coming from the ovum. So these two, when they combine, they form a zygote with allele combination of capital B and small b. Here we can see that it is heterozygote, or it is a heterozygous. Therefore, these two cannot express themselves together. One of them is going to dominate and another one is going to be suppressed. So which one is dominant? When you look at them, capital B is dominant and small b is recessive. And what instruction is given by capital B? Capital B is giving an instruction that this offspring should be having a coat color of black. And small b is giving an instruction that this animal should have brown color. So which one is going to express itself? So you will see that here, because capital B is dominant, capital B is going to express itself, and smaller b is going to be suppressed. Therefore, the offspring is going to be having a black coat color. Now, that is when we are using this sperm. It has got a chance to combine with this one, and it has also combined with that one. So now we are going to go to our second sperm. What if it is this sperm we are looking at? This sperm can either go with this egg here, and when this one goes with this egg, therefore they are going to end up producing this zygote here. If they produce this zygote has got two alleles, small b allele and capital B allele, which indicates that the offspring is going to be coated black because there is a dominant capital B allele which instructs the color to be black. Then again, this same sperm can again have an opportunity to go with this ovum here. When it goes with this ovum here, then they are likely to produce this offspring here. And when they produce this offspring here, this zygote here is going to have two alleles. And these alleles are small b allele and another small b allele. And these are homozygous, meaning that they express themselves in the same way. They have the same effect on the offspring, giving instructions that the offspring should be brown in color. So this one now is going to give us a brown colored coated mouse. So those are the possible offsprings we are likely to get when we have two heterozygous or heterozygotes being mated together. A neat way of working out the possible combinations is to use a Punnett square. It can be easier for us to work out a neat work when we use a Punnett square. This is our Punnett square. These lines, we draw these lines where we are going to put our gametes, and then we place now our gametes. Now the gametes are going to be placed in this corner. These are the male gametes. We shall place them in this corner. And then the upper corner, this is where we are going to put our female gametes. After forming our gametes by meiosis. But remember, we said that these are two heterozygotes. The male gametes will be capital B, small b. And the female gametes will also capital B, small b. So this one can go with this one. And they are going to form this gamete here, which is capital B and capital B. Then this one can go still with this one, and they will form capital B and smaller b. Now we have this one can go with this one. So we cross like this and this one here. So we shall have now here capital B and smaller b. Again, we can have this gamete here going with this gamete here. So there they will produce small b and smaller b. So those are our offsprings. These are our offsprings that we have formed. These ones under black, they are our offsprings. There is an offsprings with capital B, capital B. This one is going to have a black coat. We have another offspring, capital B, small b, which is going to have a black coat. We have another one, capital B, small b, which is having a black coat. And then we have another one, smaller b, which has got a yellow coat color. These are now what we call the F2 generation or second filial generation. You draw the grid. What we do, we draw the grid as we have seen. Our grid is that one that we draw there. That is our grid. Enter the allele in the gametes and enter the possible combination. So you, after drawing the allele, the grid, then you enter the gametes. These are our gametes. You enter the gametes here, and here you enter the gametes like the way we have done it, all of us have seen. Then you enter the female gametes in that corner. And then after that one, you carry out the combination. This one can go with that one. And then this one can go with this one. Then this one can also go with this one. Then this one can go now with that one. So those are the combinations that we are going to form. Different offsprings of different combinations. The offsprings are in the ratio of 3 black and 1 brown. So when you look at our offsprings, which we have got here, 3 of them are going to be black. One will be black, and will be black, and the last one will be brown in color. So which means we have 3 blacks and we have 1 white, 1 brown. The 3:1 ratio is sometimes called Mendelian ratio, after Gregor Mendel, who lived between 1822 to 1846. He was an Australian monk with an interest in plant breeding and mathematics. He made experimental crosses, particularly of pea plants, to see how characteristics were inherited. So Mendel, when he was doing his experiments, he used pea plants, and he wanted to see how these pea plants, how the characteristics were being carried from the parent to the offspring. He realized that certain characteristics disappear in the first generation. For example, let's take a look here. Here, he had green pod pea plants, and he was going to cross them with yellow pod pea plants. The green pod, they have capital G as the genotype or as their allele. Capital G is the allele for the green pod. Small g, these are the allele for the yellow pods. That's why when you look at the genotype of the green pods he used, they were homozygous capital G, meaning that they were having a pair of allele for green pods. And the yellow parents, he used a yellow pod, which is the parental phenotype. The parents' pod were yellow, but the parents' genotype was small g, which is homozygous also for color of yellow. So they were homozygous allele small g. Now when you form the gametes for the green pod, they will give you capital G because both gametes are the same. They contain the same allele, capital G. And the gametes for the yellow pods are small g. Both of them, they contain small g. So we shall put our gametes in the Punnett square. Have the gametes here for yellow and then gametes here for the green. So these gametes, now when you cross them, we shall get the genotype of the F1 offspring, which is capital G, smaller g. This one is now heterozygous. Now, and it is the offspring, F1 offspring genotype, capital G, small g. Now, this capital G, small g is going to give us all the plants having a green pod. Why? Because the capital G is dominant to the small g. Therefore, capital G is going to express itself, and then all the offsprings will have the green pod. The offspring phenotype is now green pods. That's what he got. Now, when you look, all these are green, but the parents, one of the parents was yellow. So that's why Mendel realized that certain characteristics, they disappear in the first generation, F1 generation. So you find that the yellow offsprings have disappeared. Yet we use the gametes from the yellow parent and the green parent. But one of the characteristics, which is the yellow offsprings, all of them disappeared. They did not appear in F1 generation. All the offsprings were green in color. One character had disappeared in the first generation. They could reappear in the second generation. And so he developed the idea of dominant and recessive characteristics. So when Mendel was continuing to cross, he found out that when he crossed the first filio generation, the characteristics that disappeared in the first generation reappeared in the second generation. How did he do it? Now, the F1 offspring phenotype, all of them were green. And all of them, they have a genotype of capital G, small g. So he brought them and crossed them with their green pods, which is their phenotype, and their genotype was capital G, small g. So when they formed a gamete, each one formed the two gametes, capital G and small g, capital G and small g. Then he placed the gametes in the Punnett square. When he placed them in the Punnett square, he got now the F2 offspring genotype, which is giving us a new or the reappearing of the character which disappeared in the F1. You remember in F1 offspring, all of them were green, capital G, small g. All of them were green. But now when we crossed the F1 to give us F2, the character which disappeared now has reappeared. So now we have capital G as one of the genotype, capital G, small g, capital G, small g. All these ones are green in color. But one of them, which is small g, is yellow in color, meaning that it has appeared in the second generation, but it disappeared in the first generation. So that is what Mendel observed, that the character which disappeared in the first generation reappears in the second generation. The recessive characteristics were suppressed in the first generation. So these recessive characters were suppressed in the first generation. Remember, in the first generation, we got small g and we also got capital G. Now the capital G has got a dominant character and small g has got a recessive character. So when the two comes together, a dominant and a recessive is going to be suppressed, meaning that the recessive will not express itself because the capital G is for green color, small g is for yellow color. But when the two come, they give out one color which is the green pod color. Where is the instruction for the small g? The instruction for the small g has been suppressed. Why? Because capital G is dominant over small g, which is recessive. So the recessive is going to be suppressed and the dominant is going to express itself. That's why all the offsprings were green in color, but they had a gene for brown color, which was suppressed. But when the two were crossed, the F2 generation, the brown color reappeared. Why? Because in this combination, we had a combination of small g, recessive alleles aligning themselves together and both of them expressing themselves, and then that character reappeared. His second generation process involved a single characteristic, produced a ratio of approximately 3:1. So when you look here, you will see that the F2 generation, we have, these are green. All of these ones are green. And how many are they? They are 3 in number. And only this one is brown. This one is brown. And how many are they? It is 1. So it gives us a ratio of 3 green and 1 brown. From these results, he drew accurate conclusion about the mechanism of inheritance. Although he knew nothing about the chromosomes and the genes at this time. So he made his conclusion about inheritance mechanism, even though he didn't know much about chromosomes and genes at the time he did this. Although capital B and capital B small b mice look identical, the capital B small b mice are not true breed. They don't breed true. Now, when you look at this rat here, this rat has got a genotype of capital B. And when you look at all these rats we have got here, all these rats have got a genotype of capital B small b, which means that capital B, which is this one, is a black mouse, looks identical with capital B small b. But the capital B small b, these ones, they will not breed true. Meaning that when you cross them, they will produce some marks which are brown in color. When mated together, there is a chance of 1 in 4 of their offspring will be brown. So when you get this F1 generation, the heterozygotes, which is giving us black offsprings, if you mated them, you get 2 of them and mate them, they will give you offsprings which will have 1 out of 4 offsprings being brown in color. So we say they do not breed true. This is also a chance because sperms and ova meet at random will be brown. So this combination, this one going with this one is random, or again this one going again with this one, it is also random. It can also be this one going again with this one, and we shall get, if this one goes with this one, we shall get this offspring. Or it still can be this one going with that one, and when they go with that one, it can give us this offspring. It is random. It is not planned. It is random mating. Therefore, there might be brown or not there. The litter of 5 may contain no brown mice. In a litter of 12, you might expect 3 brown mice, but you would not be surprised at anything between 2 and 5. So we can cross and we get litters of a ratio of 1 out of 4 being brown, but it can also be 5 individuals in a litter without a brown one, and all of them are black. When we have 12, we would expect a litter having 12 to have at least 3 brown mice. But you should not be surprised to find there between 2 to 5. The total offspring from successful mating of the heterozygotes would be expected to produce something close to 3:1 ratio. So when we cross a heterozygote with a heterozygote, we are likely to have a ratio of 3 blacks and then we have 1 brown, which we call Mendelian's ratio. So when we cross the heterozygotes, which are capital B, small b, capital B, small b, we are going to get a ratio of 3 blacks and 1 brown. For example, 6 successful litters might produce 35 black and 13 brown mice. This is a ratio of 2.7 to 1, which is nearly enough to 3:1. 3 blacks and 1 brown. The offsprings of the zygotes are the F2 generation. So these are our heterozygotes. When we cross the heterozygotes, the offsprings we get here, all these offsprings here, are the ones that we call the F2 generation offsprings. The genetic constitution of an organism is called its genotype. So here, when you come here, for these ones here, the heterozygotes, this is their genotype, capital B, small b. Those are their genotype. But when you come to these ones now, this one, the genetic composition or genetic constitution is capital B, small b. That is its genotype. This one's capital B, small b, capital B, small b. These ones, that is their genetic constitution. And this is small b, which is its genetic constitution. The visible or physiological characteristics of an organism are called its phenotype. Those are what is observable, or the physiological characteristic, that one which is inside, which is not seen, of an organisms are what we call the phenotype. For example, the appearance of this rat is black. Therefore, its phenotype is black coated. And this one is also black. Therefore, its phenotype is black coated. The phenotype of this mouth is black, and its genotype is capital B, capital B. So we are saying this one here, its phenotype is black, what we are seeing. But the genotype, genetic constitution, is capital B, capital B. This one is what is inside, which we are not seeing. It's what we are referring to as the genotype. Its genotype is capital B, capital B. Its phenotype, what is visible, is black. The phenotype of this mouth is also black, but its genotype is capital B, small b. So when you look also at this mouse, you will see that its appearance, which we call the phenotype, is black. But its genotype, which is the genetic constitution, is capital B, small b. The phenotype of this mouse is brown. Now, when you look at this one, what we are seeing is the phenotype, and that phenotype is brown, but its genotype is smaller b, smaller b. So when you look at its genotype, which is not seen, or what we call the genetic constitution, is smaller b, small b. These tobacco seedlings are the F2 generation from a cross between heterozygous, capital C, small c, parents. Now we have tobacco seedlings here. You can see that some of them are green, others are white, meaning that those ones that we are seeing with white, they don't have chlorophyll. Those ones that we are seeing which are green, they have got chlorophyll. Capital C is the gene for chlorophyll. Capital C is the gene for chlorophyll. Meaning that if we have heterozygotes, and since this one is F2 generation, if it is F2 generation, it means it comes from F1 generation. And the F1 generation, they always have, or they are always heterozygotes with heterozygous allele, capital C and small c. So, meaning that where we have capital C, it means that that seedling will have chlorophyll. And the one which has got small c That is an allele for no chlorophyll. So CC, small c plants can make no chlorophyll. So these plants that we are seeing white in color, those are plants which don't have chlorophyll. And therefore it means that their genetic constitution is small c, meaning that they don't have chlorophyll. There are 75 green seedlings. All of them, they make up 75 seedlings. What is the ratio of green to white seedlings? What is the ratio of green to white seedlings? So which one has green and which one has white seedlings? What is their ratio? What ratio would you expect? What ratio would you expect? So which ratio are you going to expect here? The ones which are green and the ones which are yellow or white. There are 21 white seedlings. This is a ratio of 75 to 21 or 3.57 to 1. That is the ratio. So when we get our Punnett square and we draw the grids, after drawing the grids, then we put in our gametes. And since the gametes are coming from heterozygotes, we shall have capital C and small c, capital C and a small c. Then we carry out the cross. When you carry out the cross, you will find that 3 out of 4, they are going to be green in color. And it is going to be only 1 out of 4 which is going to be white, forming a ratio of 3 to 1. There is one capital C, which is this one. There are 2 capital C, small c. We have 2 capital C, small c, and then this one capital C, small c. And there is only 1 small c, We have this one, small c, only 1. A ratio of 3 green to 1 white seedling. So we have 3 green seedlings and only 1 white seedling. Is 3.57 to 1 near enough to 3.1? 3 to 1? That's a question. Is 3.57 to 1 near enough to 3.1? You would expect the cross to produce 72 green to 24 white seedlings, which gives us a ratio of 3 to 1. In most population of animals, there are approximately equal numbers of males and females. This is a result of a pair of chromosomes, the sex chromosomes, called X and Y chromosomes. So when you look here, we have X and Y. These are sperm mother cells. And here we have X and X, which are ovum mother cells. So we have this one is a male because he has X and Y allele or chromosome, and this one is a female, X and X, because it has got two chromosomes, X and X. The X and Y chromosomes are homologous pair, but in many animals, Y chromosome is smaller than the X chromosome. In many animals, the Y chromosome is very small compared to the X chromosomes. Females have two X chromosomes in their cells, and males, they have one X and they also have one Y in their cells. So this one is for the female, for the male. They have one X chromosome and Y chromosome. Whereas this one here is for the female because they have two X chromosomes in one cell. Females have two X chromosomes in their cells. As we have seen, they have got two X chromosomes in their cells. And the males, they have one X and one Y chromosomes in their cell. As you can see here, one X and one Y in their cells. After meiosis, the sex chromosomes are separated. So the gametes receive only one, either an X or a Y. So during meiosis, we end up getting gametes, and each gamete has got one chromosome. For example, this sperm has got the X chromosome, and this one has got the Y chromosome. But when you go to the female, for the female after meiosis, we shall have ovum, each one with one chromosome, and each one will be having X chromosome, X chromosome. This shows that there is a 50% chance of a boy or a girl baby. So if we have the father producing X chromosome and Y chromosome, and the mother produces X and X chromosome. So when we get the combination, we are likely to have 2 female individuals, and there is a possibility of having 2 male individuals. So the ratio is 1:1, which makes it 50% a boy, 50% a girl. Because this ratio depends on the chance of meeting X and Y gametes, and human families are small. The ratio 1:1 is rarely seen, even in large families. A family with 6 boys may hope for a girl next time, but there is still only 50% chance of getting a girl. In the population as a whole, the 1:1 ratio holds good. Very few human characteristics are controlled by a single gene. Characteristics such as height or skin color, these are controlled by several genes acting together. Those characteristics which are controlled by a single gene are usually responsible for inherited defects. Cystic fibrosis, it is recessive. The glands of the alimentary canal, these ones produce thick mucus, which affects breathing, digestion, and susceptibility to chest infection. Achondroblastic dwarfism, this one is shown by dominant. The head and the trunk, they grow normally, but the limbs, they remain short. Albinism is a recessive. Albinos cannot produce pigments in their skin, hair, and even eyelids. Polydactyl, it's dominant. Here is where an extra digit may be produced on the hands or feet. So these are some of the conditions which are controlled by a single character or a single gene. They mainly result in defects. In genetic counseling, if the genotypes of the parents are known, it is possible to calculate the probabilities of their having affected child. So, if the genes of the parents are known, it is very easy to calculate their probability of having an affected child. For example, a male achondroplastic dwarf marries a normal woman, what are the chances of them having an affected child? We know achondroblastic dwarfism is dominant. So we would expect that male to have these genes. Capital D, small d. Not capital D, because capital D is not viable. Those ones do not survive. So if a woman is normal, then we are going to have these are the genes for the woman. So when we cross, we are going to get their offsprings. Two of them will be capital D, small d, meaning that there will be a chondroblastic dwarf. And the other two will be small d, meaning that they are normal. So the chance of having an affected child is a half, or which is 50%. The father's genotype must be capital D, small d. Why? Capital D is not viable. An individual or a zygote that is formed with capital D does not survive, cannot be produced. The mother must be small d, since she is not a dwarf. There is a 50% probability of them having an affected child, as we have calculated. That this is the genotype of the parents. These are the genotype of the mother. These children here are going to be affected. And these children here will be normal, which makes it 50% children can be affected and 50% can be normal. What are the probability if both parents are affected? If both parents were affected, what do you think would be the probability? That means that now the lady will also be capital D, smaller d. Therefore, here we are likely to have capital D. This one is not viable. This one will be capital D, smaller d. And here we are likely to have capital D, smaller d. And then here we are likely to have smaller d. So if this one is not viable, this one cannot be produced, there is a chance of 2 out of 3 children being affected. That is 2/3 of their children will be affected. This one will be affected. This one will also be affected. And this one will only be the normal child. The incidence of achondroplasia is about 1 in 30,000. These cases arise as a result of mutation. The affected children are born to normal parents, but it is a result of mutation. A mutation is a spontaneous change in a gene or in a chromosome. Mutations are almost harmful. Cystic fibrosis is recessive. If two normal parents have an affected child, they must both be heterozygous, capital N, small n, for the recessive allele, small n. So if we have this child being produced with cystic fibrosis, it means that the parents, both parents, were heterozygous for the recessive allele, small n. That is when they are able to produce a child who is cystic fibrosis. An NN parent would have cystic fibrosis. An capital N parent would produce only normal children. Since the parents are known to be heterozygous, it can be predicted that their next child has a 1 in 4 chance of inheriting a disease. The parents are heterozygous. It means one of the four children is likely to inherit the disease. That is, if the parents are heterozygous. This chance applies to all subsequent children, all children that will follow. There is that chance that 1 out of 4 will be having or will inherit a disease. Even if the parents have 5 normal children, there is still a 1 in 4 chance of the next child being affected. They may have all their children normal, maybe 5 of them, but every time they are going to produce another child, there is that 1 out of 4 chance that that child may be affected. If the heterozygotes could be detected before they had children, they could be counseled about the likelihood of having affected children. Families at risk, for example, cystic fibrosis in a relative, may be offered DNA testing to look for 1 or more genes which cause the disease. Those are for families at risk. If there is someone, a relative, with cystic fibrosis, they can carry out DNA tests to look for 1 or more genes which can cause the disease. Sickle cell anemia. This is also recessive. HB This represents hemoglobin. HbA is the allele for normal hemoglobin. HbS, this is the allele for sickle cell hemoglobin. So if we have parents who are carrier, meaning that they have a recessive allele for sickle cell anemia, and if these parents produce children, this child is going to be normal. This child is a carrier, and this one is also a carrier. Then the last child here is a sickler. So there is a possibility of them having 1 out of 4 being a normal child, the possibility of having 2 out of 4 being sicklers, or being carriers, and there is a possibility of them having 1 out of 4 being a sickler. A person with the genotype HbS will suffer from sickle cell anemia. This person here, who is having a genotype of hemoglobin sickle, is going to suffer from sickle cell anemia. And a person with the genotype HbA and HbA is normal. So this person here is going to be normal. The genotype HbA, HbS, this one produces sickle cell traits because Hb normal is incompletely dominant to Hb sickle. So this individual here will produce sickle cell traits because the Hb normal is not completely dominant to Hb sickle. The heterozygote HbA, HbS has few symptoms, but he or she is a carrier for the disease. They have few symptoms, but they are carrier for the disease. Heterozygous recessive individuals do not usually exhibit any disease symptoms because their offsprings may inherit the disease. The heterozygotes are called carriers. These heterozygotes, we are saying they are individuals who do not exhibit the disease symptoms, but their offsprings may inherit the disease. So the heterozygotes, we refer to them as carriers. Similarly, individuals with the genotype capital N, small n, they are also carriers for cystic fibrosis. It is sometimes possible to work out the genotypes of the parents and to track the inheritance of an allele by studying the family trees. If we take this one to be a normal female, this one to be a normal male, this one affected female, and here we have a affected male. If we have the parents, normal male and a normal female, parents have a normal phenotype, but they produced one of the child being affected with the disease or the effect. For this to happen, both parents must have been heterozygous genotype, which is capital N, small n, for the characteristics. So if this one was to appear, the parents were heterozygous, capital N and small n. This means that there is a possibility of having small n with smaller n child who is going to be affected. If one of the parents is homozygous for a dominant allele, all the children will be affected. That is, if that effect is as a result of a dominant allele, and one of the parents has got a dominant allele, that means that all the children will have that dominant allele, which causes the effect, and those children are all going to be affected. If one parent is heterozygous for a dominant allele and the other is homozygous recessive, there is a chance that half of the children are going to be affected. So this character expresses itself in the presence of a dominant allele. So if one of the parent is heterozygous for that dominant allele and the other one is homozygous recessive, then two of their children, half of their children are going to be affected. If both parents are heterozygous for a recessive allele, there is a chance that 1 in 4 of their children will be affected. So there is a possibility that one of the child will inherit the recessive allele. And when that child inherits the recessive allele, that child is going to be affected. What can you deduce about the genotypes of the grandparents from this family tree? We have cystic fibrosis. Cystic fibrosis is a recessive, small n, smaller n. And when you look at the parents, the grandparents are normal. Even the children are normal. But one of the grandchild is affected by cystic fibrosis. So what do you deduce about the genotype of the grandparents from this family tree? Cystic fibrosis is caused by a recessive gene. An affected person must therefore have the genotype small n, small n. Since neither of the grandparents is affected, they must either be capital N or capital N, small n genotype, because they are normal. If they were both capital N, none of their children or grandchildren could be affected. That is, if they were capital N, they would not have any of their children affected by cystic fibrosis. That is, if they had capital N, If one was capital N, small n, and the other capital N, then there is a chance of about 50% of their children could be a carrier capital N, small n. That is, the grandparents are normal, but one is capital N, small n, and the other one is capital N and a capital N. There is a chance that 50% of their children are going to be carrier, meaning that they will be having capital N, small n, and their children could be capital N, small n. That is 50%. If one of the carriers marries another carrier, there is 1 in 4 chance of them having affected child. So if we have this one is a carrier and this one was also a carrier, there is a possibility of 1 out of the 4 children being affected. The genotype of the grandparent must either both be capital N, small n, or one is capital N, and the other is capital N, small n. For this condition of cystic fibrosis to appear in their grandchildren. Hello, thank you very much for participating in Heredity. Please do like, share, and subscribe.